All right, hi, my name is Catherine Leong, and today I will be sharing work from the Neumeier lab looking at gene expression changes in serotonin neurons in a mouse model of acute stress. So this is a great time to be sharing this data as we just published our manuscript in molecular psychiatry, and almost all of the figures I will be sharing with you today come directly from this manuscript. So we were interested in how acute stress affects gene changes in serotonin neurons. And so we subjected young mice to a model of forced swim that has been previously validated as a model of acute stress in mice. And we were interested in the serotonin neurons, and so we used a PET Cree crossed with ribotag transgenic mouse to allow us to look at gene expression changes specifically in serotonin neurons in these mice. And this is really the most important reason why we did this, because we know that serotonin is important. And, you know, in psychiatry, a lot of our most important drugs target the serotonin system, and polymorphisms of serotonin-related genes are associated with stress-related disorders. But yet, it's really grossly understudied. Up until recently, this is understandable. It's pretty technologically challenging to do so. And the raphe are really small, and even when you microdissect the raphe, it's really hard to isolate the serotonin neurons, especially when you have so few cells to begin with. You know, but new technology is making it more feasible for us to look at the serotonin neurons, and we should be taking advantage of this. So here I'll be explaining how we were able to look at what was happening specifically in the serotonin neurons at a transcriptional translational level in response to stress. So after this acute stress, we rapidly dissected the brains from these animals and took a dorsal raphe punch. And then we extracted mRNA from the dorsal raphe, and this is a heterogeneous population of mRNA from all the cells in the raphe punch that we took, and it's, you know, mRNA from both serotonergic and non-serotonergic cells in the dorsal raphe. And then we divided the homogenate into two fractions. We reserved a portion of the original heterogeneous homogenate into a portion called what we called the input fraction. And then we incubated the rest of the mRNA with anti-HA magnetic beads. And with these magnetic beads, it allowed us to immunoprecipitate the mRNAs that were actively being translated by the ribosomes that had this HA tag. And if you remember from my initial slide, we used Cree ribotag mice. So these, or PET1 Cree ribotag mice. So these HA tags are specifically expressed in the ribosomes of serotonin neurons in these mice. And so with the pull down, we're able to isolate mRNA that's actively being translated by the ribosomes in these serotonin neurons. And so with this, we were able to look at the gene expression changes in both serotonergic and non-serotonergic cells, and use a lot of computational filtering to filter out the noise and come up with you know, these gene expression changes, both in the input fraction, which again was that gene expression changes in that heterogeneous population of mRNAs from serotonergic and non-serotonergic cells in the dorsal raphe, as well as changes in the translatome of serotonergic neurons in response to stress. And if we exclude the mRNA changes in the non-serotonergic neurons and really only look at the changes in the translatome in the serotonergic neurons, and if we can look at the gene expression changes that are common to both male and female mice, it came down to two different genes that were upregulated in response to stress that passed our computational filtering. And of these two, we decided to focus on Fkbp5 for a few different reasons. You know, Fkbp5, when we searched the literature, we found that polymorphisms of Fkbp5 have been found to be associated with psychiatric illness such as depression and PTSD, as well as psychiatric outcomes such as suicide. Now, if we look at a molecular level, we saw that Fkbp5 has known mechanisms for regulating the glucocorticoid signaling at a molecular level, including decreasing trafficking of the receptor to the nucleus, as well as decreasing the affinity of glucocorticoids for the glucocorticoid receptor itself. So in essence, an increase in Fkbp5 results in a sort of glucocorticoid resistance. Okay, so if we look at the expression of Fkbp5 in slices of dorsal raphe pre and post stress, you can see that Fkbp5 is expressed in serotonergic and non-serotonergic neurons, Fkbp5 in green and PET1 in red. And then when we stress the mice, the expression of Fkbp5 greatly increases in PET1 or serotonergic neurons, as you can see with a huge increase in the yellow overlap here. And if we quantify the expression of Fkbp5 in these neurons, you can see that the percentage of serotonergic neurons with Fkbp5 expression significantly increases following stress, and that the intensity of the Fkbp5 signal in serotonergic neurons also increases after stress. And and I should say that we also looked at expression changes of mRNA using qPCR as well as protein, and we saw the data isn't shown here, but for time's sake, but the data correlated with the results of this gene expression, which was done using RNAscope. Next, we were interested to see what would happen if we blocked Fkbp5 and what would happen with mouse behavior. And so we used a pharmacological inhibitor of Fkbp5, SAFET2, which has previously been used in other parts of the brain to investigate Fkbp5 function, but has not been looked at in the dorsal raphe prior to this. So we looked at a couple different behaviors, including sucrose licking, and we saw that microinjection of SAFET2 into the dorsal raphe, we looked at water licking and sucrose licking, and you can see that vehicle-injected animals, as well as the SAFET-injected animals, didn't show much difference in water licking as a control. But then when we look at sucrose licking, keeping in mind that the y-axis is 10 times greater in the sucrose licking graph compared to the water licking graph, you can see that the vehicle-injected animals showed no change in sucrose licking behavior, but that following stress, there was a statistically significant increase in sucrose licking following exposure to stress when we block Fkbp5 with SAFET. Now, this was interesting because it showed a decrease in the anhedonic behavior in this sucrose licking model of anhedonia. We also looked at immobility and saw no change in immobility when we compared SAFET-injected animals to vehicle-injected animals. And then if we actually quantify sucrose, the change in sucrose licking after stress in SAFET-injected animals compared to vehicle, there was a statistically significant increase in the change in sucrose licking. And if you look at sucrose licking behavior over time after acute stress, you can see that there's a pretty rapid increase in sucrose licking after stress if you block Fkbp5 in the dorsal raphe, whereas vehicle-injected animals had a pretty slow increase in this sucrose licking behavior. So in summary, we were interested in looking at gene expression changes after exposure to acute stress, specifically in the dorsal raphe, and we used ribotag technology to be able to look at gene expression changes specifically in serotonin neurons. And specifically actually the translatome changes, because we were able to look at mRNA that was actively being translated in serotonin neurons. And with a bunch of computational filtering, we were able to narrow it down to 2 genes that changed specifically in serotonin neurons following stress. And we decided to focus on Fkbp5 because of its link between these serotonin neurons and glucocorticoid signaling. And we found that blocking Fkbp5 with a pharmacological inhibitor resulted in a behavior change linked to anhedonia. Now, in conclusion, we've shown that there is a mechanistic link between stress and glucocorticoid signaling in serotonin neurons, and that when exposed to stress, the system moves toward glucocorticoid resistance by an upregulation of Fkbp5. And blocking Fkbp5 with a pharmacological inhibitor, SAFIT, resulted in a decreased anhedonic behavior as measured by an increase in the sucrose licking following stress. So moving forward, it will be interesting to continue work to connect the dots between psychiatric illness, stress, and stress hormone signaling. I should say one last thing I meant to mention was that we also have looked, we are currently looking at blocking Fkbp5 specifically in serotonin neurons, as the SAFIT is a pharmacological inhibitor microinjected into the dorsal raphe, but not specifically targeting serotonin neurons. And we are currently looking at serotonin neurons specifically using CRISPR, and those experiments are ongoing. So continuing to look at connecting the dots between psychiatric illness, stress, and stress hormone signaling, there's a lot of questions that remain to be answered. I think the most interesting would be looking at whether glucocorticoid resistance in serotonin neurons is part of this mechanism behind the pathological development of stress-related disorders. And on the more clinical side, maybe could Fkbp5 polymorphisms predict your response to stress? And can we find out more about this interplay between psychiatric illness and hormone signaling to find biomarkers that would be that could predict your response to stress, or even testing of hormone levels that could predict psychiatric outcomes? And finally, can we manipulate parts of this pathway for drug development? And with that, I'd like to thank all of the members of the Neumeier Lab, and especially my co-authors, Kevin and Adam, and our PI, John Neumeier. And our funding is listed here as well. Thank you.